This module is about factorial ANOVA, analysis of variance with two or more independent variables. Thus far we've talked about analysis of variance in post hoc tests with one independent variable, but you can have 2, 3, as many as you want. When we're done with this module, you should be able to answer these questions. What are the main effects in analysis of variance? What are interactions in analysis of variance? How do you know when you have one? What does it mean for a design to be completely crossed, balanced, and orthogonal? You should be able to describe each term in a linear model like this one. Y sub ijk is equal to mu plus alpha sub j plus beta sub k plus alpha beta sub jk plus e sub ijk. You should also be able to look at an analysis of variance summary table and identify mistakes. You'd be surprised how often people make mistakes in these things, and a little checking you can make sure that that doesn't happen to you. You should be able to find critical values of f from a table for a given design. You already know how to do that for one-way analysis of variance. It's a little bit slightly more complicated for two-way or three-way. You should be able to say how post hoc testing for factorial ANOVA differs from post hoc testing for one-way. And you should be able to describe a concrete example of a two-factor experiment and tell me or other people why it is interesting or important to consider both factors in one experiment. Okay, so the basics, the nomenclature of analysis of variance with two independent variables. We've had one-way ANOVA so far. You can have two independent variables, three, as many as you like. Independent variables for no good reason are called factors in analysis of variance. This is not the same thing as factors in factor analysis. Factor means independent variable in analysis of variance. So the simplest kind of design, remember design is a data collection strategy or method or process. Simplest design we can have is two independent variables, and each of them will have two levels. In our example, we're going to be teaching research methods to undergrads, and we're going to test them all on an exam to see how well they learn, and we're going to assign each person to one of four conditions. Independent variable one, workbook or not. So some people will get a workbook to help them study, and some people will be left to their own devices, as they usually are in studying for an exam. And some people will be given a cup of coffee and others will not. So we're going to have to do something tricky to see. Randomly assign people to that condition and make sure that people don't cheat and get lots of caffeine. So the two variables are the presence or absence of coffee and the presence or absence of a workbook. It's a very simple design. We have four possible conditions. Each person can get one of each of the two independent variable levels. So we have some people who get no workbook but do get a cup of coffee. They're the caffeine-only group. We have others who get the workbook and get coffee. They have both. Then we have the workbook only, and then we have the poor people who are left to their own devices entirely, and they're the control group that has neither coffee nor the workbook. Well, I suppose that we did that study, and we had 30 people. We've got about 120 people in our class, and we assigned them randomly to one of these four conditions, and we got 30 people in each condition. And we get these kinds of results. The caffeinated group has a mean of 80 on our exam, so it's one of those percentage exams from 0 to 100. They got a mean of 80 and a standard deviation of 5. The people who got both have a mean of 85 and a standard deviation of 5. People who got neither have a mean of 75 and a standard deviation of 5. And the people who got the book but no caffeine have a mean of 80 and a standard deviation of 5. Notice that if we take the grand mean over everyone, we get 80. So the average for the class is 80. If we look across the rows or across the columns, we can see what happens with one factor ignoring the other factor. So if we look at what happens with people who didn't get a workbook, ignoring whether or not they got caffeine, those folks average 77.5. People who got the workbook, went across both those, they're at 82.5. And similarly, we can average across the columns. So for the first row, these are the people who got the caffeine. If we forget about whether or not they got a workbook, those people have an average of 82.5. All right, so there's our cell means. A cell is a treatment combination. So one cell is the caffeinated group. They got no workbook, and yes, they got a cup of coffee. So that's called a cell or a treatment combination. And you can see we have 4 of those, and then we have 2 row means, 2 column means, and 1 grand mean. I'm going to graph the interaction using lines. Some people will tell you that you shouldn't actually use lines, but I don't care. I think it's very clear what you got. So I've got 2 lines, one with the caffeine and one without the caffeine, and then I've got 2 edges of the graph that stands for no workbook and workbook. And you can see that the lines slant from lower left to upper right, so that the lines are higher on the workbook yes than the workbook no, and the caffeinated group is higher in the graph than the decaf or non-caf group. Probably should have switched the red and green lines around so the caffeinated group got red and the decaf got green, but whatever. The graph shows the means for the groups on the left. So it goes from 75 on up to looks like 86 on that line. Okay, so when we're interpreting these graphs, we can see main effects and interactions. So with a graph like this, the main effects will be shown by slopes. So does it slant from left to right or right to left? The lines, do they slant? And are there a break between the lines? So that'll tell us about the main effects. An interaction between our 2 independent variables, that is an interaction between workbook and caffeine, would be seen by a lack of parallelism in our 2 lines. And I should tell you that interactions are one of the main reasons that people actually do factorial ANOVA. We want to investigate whether the impact of one variable depends on the value of another variable. So does the effect of caffeine depend on whether or not you have a workbook? Does the effect of a workbook depend on whether or not you have caffeine? That's the investigators really want to know that. Otherwise, they could do the studies independently. You could have a workbook study, you could have a caffeine study, it could be done more quickly, more easily, but if these 2 things do interact, caffeine and workbook, then the only way you'll know that is by doing this study with both factors at once.